Hello everyone, welcome back to Zoology with Rutuparna. In today's class, we will learn about the five kingdom classification proposed by R.H. Whitaker in 1969. This classification is one of the most important topics in zoology and in your exam point of view. So let's begin. Initially, scientists classified living organisms into only two kingdoms, kingdom Plantae and kingdom Animalia. Then why was a new classification needed? See, this system had many limitations. For example, bacteria are neither plants nor animal, so in which kingdom we will place it? So this is a limitation of two kingdom classification. Another example is fungi. So they cannot prepare their own food. So they are not plants. And then third, euglena. Euglena shows both plant and animal characters. Then where it will be placed? So unicellular organisms differ greatly from multicellular organism. Therefore, a better classification system was required. Then the scientist R. H. Whitaker proposed five kingdom classification in the year of 1969. So those five kingdoms are Monera, Protista, Fungi, Plantae and Animalia. You can see the picture present over here. Monera are unicellular, means they are made up of a single cell. It has no true nucleus, so it is nucleated and no presence of membrane bound organelles. Cell wall present and that is usually made up of peptidoglycan. Peptidoglycan is a polymer made up of sugars and amino acids and it consists of NAG and NAM. NAG means N-acetyl glucosamine and NAM N-acetyl muramic acid. So these sugar molecules are joined by beta-1 for glycosidic bonds. If you see the reproduction, then it is generally asexual in the process of binary fission. And the example is bacteria. Okay, one question. Is virus coming under the kingdom Monera? Type a comment on this question in chat box. Okay, let it. Viruses are not coming under any of the five kingdom classification because they are acellular, lack independent metabolism and can reproduce only inside the living host cells. Okay, so this is the reason. Now coming to the second kingdom, Protista. See, these are mostly unicellular eukaryotic organisms because they have true nucleus and membrane bound organelles. Cell wall absent in most forms. They have various types of nutrition like autotrophic, heterotrophic or mixotrophic. And reproduction can be asexual and sexual types. Example, amoeba, paramecium, euglena, chlamydomonas and many examples are there. You can see the picture present over here. And then coming to the third kingdom, fungi. These are eukaryotic and mostly multicellular, cell wall present and that is made up of chitin. Chitin is a hard, strong, nitrogen containing structural polysaccharides and they are heterotrophic. Heterotrophic means simply they depend on other for their food and their body made up of hyphae forming a mycelium. You can see here and they reproduce by spore both asexually and sexually. For example, mushrooms, scientifically known as cucurbita, yeast, penicillium, etc. Now coming to the fourth kingdom, Plantae. These are multicellular eukaryotic organisms, cell wall present which is made up of cellulose. They are autotrophic, that is making their own food by the process of photosynthesis. Chlorophyll present in chloroplast and their food storage organ is starch. For example, moss, fern, mango, sunflower. See, Plantae is a vast term. It further classified in many categories like Bryophyta, Pteridophyta, Gymnosperms and Angiosperms. So we will discuss about this in detail later when we will study about the plant physiology. So now come to the fifth number of kingdom and that is Animalia. They are multicellular eukaryotic organisms. Cell wall is absent. They are heterotrophic, generally motile at some stage and it has a well developed tissues and organs. For example, human, earthworm, fish, insects, etc. So there are many phylums in animal kingdom. Total of 11 phylum are there. You can see here: Porifera, Nidaria, Ctenophora, Platyhelminthes, Nematoda, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. So now coming to another important point, and that is the three domain of life. See, after five kingdom classification, scientists discovered that archaea are genetically very different from bacteria, even though both are prokaryotes. Therefore, scientist Carl Woese proposed the three domain system in 1990. See, scientist Whittaker placed both the bacteria and archaea together in kingdom Monera. However, molecular studies showed major difference between them. So, scientist Carl Woese used the 16S rRNA sequencing, which revealed that archaea are genetically distinct from bacteria and more closely related to the eukarya. So, the three domains are bacteria, archaea and eukarya. Generally, bacteria are prokaryotic, unicellular, not having a true nucleus, no any membrane bound organelles, and cell wall contain peptidoglycan and reproduce by binary fission and found in everywhere. Okay, for example, E. coli, Escherichia coli, Bacillus, and Staphylococcus, Streptococcus, cyanobacteria like Nostoc, Anavena, etc. Then the second domain archaea, and that is prokaryotic, unicellular, not having a true nucleus. Cell wall does not contain peptidoglycan, so it is also known as pseudopeptidoglycan. And cell membrane contains ether-linked peptides, which is more stronger than ester bond, and they can survive in extreme environments. So these are some of the peculiar characteristics of archaea. For example, Methanobacterium, Methanococcus, Halobacterium, Sulfolobus, etc. Okay. Now coming to the last domain is eukarya. So these are eukaryotic, as it is clear with its name, and it has a true nucleus, it has all the membrane bound organelles, reproduction may be sexual or asexual, and it includes four kingdoms: Protista, fungi, plantae and animalia. Okay, so this is all about the three domain of life, where bacteria is the most primitive domain. Okay. Now, after learning about the three domains and the five kingdom, the next step is to understand how scientists classify and name millions of living organisms. You just imagine a large library with millions of books. If the books are not arranged properly, it becomes impossible to find one, right? Similarly, there are millions of organisms on earth. To study them efficiently, scientists arrange them in a systematic order called the taxonomic hierarchy. So there are 8 major taxonomic ranks: domain, kingdom, phylum, class, order, family, genus and species. So domain is the highest and broadest taxonomic rank. It introduced by Carl Woese, and it consists of 3 domains: archaea, bacteria and eukarya. And the second rank is kingdom. It contains many phylum, as I have already discussed about it, the 11 phylums, and a class consists of its related orders, and an order consists of its related families, and a family consists of closely related genera, and a genus consists of closely related species, and a species is the basic unit of classification. For example, lion. See, its kingdom is Animalia, phylum Chordata, class Mammalia, order Carnivora, family Felida, genus Panthera and species Leo. So this was about the taxonomic hierarchy. So let's take an overview on prokaryotic and eukaryotic cells. See, cells are the basic structural and functional units of life. So based on the presence or absence of a true nucleus, all cells are classified into 2 types: prokaryotic cells and eukaryotic cells. The term prokaryote comes from a Greek word pro means before and karyon means nucleus. So it means without a true nucleus. And the term eukaryote comes from U means true and karyon means nucleus. So it is true nucleus. So now come to the point of difference. First one is size. Prokaryotic cell has a very small in size, having 0.1 to 5 micrometer only, while eukaryotic cells has larger cell size. It's up to 10 to 100 micrometer. And the second difference is nucleus. Prokaryotes have no true nucleus. Nuclear membrane and nucleolus are also absent. In eukaryotes, true nucleus is present, which is enclosed by a double nuclear membrane and contains a nucleolus where ribosomes are formed. Then the third difference is DNA. Prokaryotes have a single circular chromosome. DNA is naked, means no histone protein is present, but plasmids are commonly present. Do you know what is plasmid? See, plasmid is a small circular double-stranded extra chromosomal DNA molecule which is separate from the bacterial chromosome. In case of eukaryotes, DNA is associated with histone proteins and have a multiple linear chromosomes stored inside the nucleus. Then the fourth difference is membrane-bound organelles. In prokaryotes, not a single membrane-bound organelle is present, while in eukaryotes, nucleus, mitochondria, endoplasmic reticulum, Golgi body, lysosome, chloroplast in case of plants are present. Fifth difference is ribosome. Prokaryotes have 70S ribosome which consists of 50S and 30S subunit, while eukaryotes have 80S ribosome consists of 60S and 40S subunit. Do you know why it is 70S or 80S? What it means exactly? See, A stands for the Svedberg unit. It's named after the scientist Theodor Svedberg. So, this 70S and 80S are sedimentation coefficient. It measures how fast a particle settles during the ultracentrifugation. So it depends on the ribosome size, shape and density, not just its mass. Okay. Next difference is cell wall. So the cell wall present in prokaryote is made up of peptidoglycan, and cell wall in eukaryotes may be cellulose in case of plants and chitin in case of fungi, and no cell wall present in animals. Okay. Then the cytoskeleton. Cytoskeleton is generally absent in prokaryotes while well developed in eukaryotes, which is composed of microtubules, microfilaments and intermediate filaments. It maintains the shape of the cell and helps in cell movement. Okay. Then the another major difference is cilia and flagella. See, in prokaryotes it is very simple and made up of flagelline protein. But in eukaryote it is complex, having 9+2 arrangement of microtubules. If you see the cell division, then prokaryotes show a sexual reproduction, and that is binary fission, where one cell divides into two identical dotted cells. But in eukaryote it shows sexual reproduction, means mitosis for growth and repair and meiosis for gamete formation. And see, the next difference is about the gene expression. In prokaryotic transcriptions, means DNA to RNA synthesis, and prokaryotic translations, means RNA to protein synthesis, occur simultaneously in cytoplasm. Okay, while in eukaryotes, transcription occur in the nucleus and translation occur on the ribosome in the cytoplasm. Okay, so now see the examples. All the bacteria are coming under the prokaryotes like E. coli, lactobacillus, nostoc, anabaena, mycoplasm. Okay, and rest are eukaryotes. You can take a screenshot of this. So this is another important concepts in your point of exams. So try to remember all the points of difference I have discussed. So this is the time to end today's class. In the next video, we will discuss detail about the animal classification and move forward with our syllabus. Okay, another thing I want to discuss with you that my academic background is from CHSC board. So I am comfortable both in English and Odia. If many of you would like to explanation in Odia as well, please let me know in the comments. Okay, so based on your feedback, I'll happy to make a bilingual videos so that learning becomes easier for everyone, because my main goal is to help you understand zoology in the simplest and most effective way. Okay, so thank you for your support. Before leaving the lecture, I want to say if you found this lecture helpful, then please like the video, subscribe to Zoology with Rutuparna, and press the bell icon. And yes, don't forget to share the video with your friends. And thank you for watching. Bye bye.